next speaker, who really brings the good news how to inhibit complement, is Daniel Ricklin from the Basel University. He comes from here, near.
All right. Thank you very much, Janos, and thanks a lot to the organizer of this wonderful meeting for inviting me here and introducing me to the fascinating world of nanotechnology. So you just heard I'm not a nanoscientist, but I have been working for the past 10 years on trying to think about how we can inhibit complement in a therapeutic way. And for me, this was a very interesting meeting to see, first of all, to get exposed to the nanotechnology, but also one of the first non-complement meetings where the term complement was popping up all over the place. And I think I have to thank Janos for priming the audience and this community to the implications that complement may have in nanomedicine. But so we want to talk a little bit about what we can do about it and what approaches we have in the complement field to deal with these interactions and these activities. And the good news about having this background here in this meeting is that I don't have to do a lengthy introduction into the topic, because many people asking me, so why the heck really you want to inhibit a host defense pathway like complement? After all, it's one of these good guys that coordinates with the coagulation system, contact system to protect our bodies from foreign intruders once we have breached the barrier due to injury or so on, and really tries to eliminate those pathogens that might come into our bloodstream. And usually it does so quite effectively. We already heard a bit about what complement is doing, and under normal conditions, physiological conditions, what we want to have, complement engages a variety of different pattern recognition proteins circulating in our body, and they are there to detect foreign patterns on intruders and activate this cascade that leads, first of all, to the tagging of the particle with these opsins. We just heard about C3b, which is the most important one. And once you have that on there without any regulators, then this response amplifies quite quickly, and it uses a series of effector pathways that can lead to lysis of a particle or membrane damage, can facilitate phagocytosis, and mediates all the downstream immune responses that are necessary then for the next steps. So that's what is complement doing typically. Of course, we don't want to occur that on a healthy host cells, and fortunately for us, we have regulators that keep complement in check on those. So if you look at it again more schematically, then we have this ideal case where we have a probing, sometimes even of host cells, lead to a little activation, but is quickly downregulated and is tolerated then and doesn't invoke any danger responses. On the other hand, if we have a foreign intruder like bacteria, viruses, fungi, you name it, then you have active sensing leading to much more complement activation, active activation of complement, rapid amplification of this activation, and hopefully to the elimination of the pathogen and the resolution. There are several occurrences in our body, several things that can go wrong with that system where complement is dysregulated and contributes to a variety of human diseases. And really their complement is not acting as it should. And we summarized that. I don't want to go into details, but in Nature Reviews Nephrology last year, we summarized some of these mechanisms that complement can go wrong. The peculiar thing now about nanomedicine or biomaterials in general is that complement is not doing something wrong. It's just that we expose our bodies voluntarily with a foreign intruder like a biomaterial, a nanoparticle or so on. So, and this might have consequences, as we just heard. So if you have a larger biomaterial, what can happen that you have sensing, activation, amplification of regulators, and that this is not eliminated, can be eliminated, but leads to chronic inflammation. Another opportunity is if you have smaller particles that they again are sensed, activating complement, are opsonized, amplified, and this can lead to unwanted fast elimination that you really would, it has some pharmacokinetic problems, or as we just heard several times here, that we have CARPA or other inflammatory reaction that can be mediated by complement. Maybe not primarily, but it can also be contributing to some of these effects. So what we also heard is that complement can be activated by various different ways, especially in biomaterials. There's very complex interaction of these pattern recognition receptors, and some can directly bind to the material surface, but as Dimitri just very nicely showed, that the corona, the protein layer that is on the biomaterial, might also mediate some of these effects. IgGs, for example, being it autoantibodies or absorbed on the surface, can activate the classical complement pathway, for example, but it has been shown that C3 can directly adsorb to surfaces, being conformationally activated, and start forming these convertases here. But no matter how complement is activated on a biomaterial surface, what you have is this amplification loop. Once you have these first C3b molecules down, it can then amplify this response and generate more and more of the opsonins. And eventually, these convertases also shift and start cleaving one of the other proteins of the complement system, C5, and this leads to the release of this anaphylotoxin C5a that can recruit and activate immune cells, and they also then interact and have inflammatory effects based on the biomaterial. And of course, complement doesn't act alone, but interacts and coordinates with other host defense systems and also inflammatory systems, so that can really lead to thromboinflammatory complications of biomaterials. So we probably have to stop whining about what complement is doing. We heard about what it can do, so we try to think about what we can do about complement in these situations. And the great thing, and what we have to really consider also from a drug discovery perspective, these host defense pathways, especially complement, act at the very early point in these activation cascades. So it is directly activated by the surfaces and then coordinates with a lot of downstream inflammatory pathways. So the idea is that if we act at this level quite early, then we can prevent a lot of the inflammatory complications that come down the road. So that renders it an interesting target for a therapeutic intervention. And indeed, over the past few years, there have been first complement inhibitors in the field now. So now we have the first specific complement inhibitors since 10 years. The anti-C5 antibody eculizumab, it blocks some of these effector pathways here downstream. And we also have a rather non-specific serine protease inhibitor called C1 esterase inhibitor that is used for some orphan diseases. So that's great, but this is not the solution for everything because there are two problems. One, they are really heck expensive, so some of these have been crowned as the world's most expensive drugs with some $500,000 per year per patient for the indications they're advertised. They're also, of course, not advertised for any nanoparticle treatment or nanomedicine treatment, but for some orphan diseases. But this is just one of the problems. The other is that they're both act at rather peripheral stages in specific initiation pathways or down at the generation of very specific effector pathways. But especially in cases where complement is involved by different means, we need to have something that acts at different levels and the more central levels. Fortunately, there have been a lot of effort in the academia and industry to improve the situation, develop new complement inhibitors. What we in our lab, together with John Lambris in Philadelphia, have been looking for is to inhibit at this very central step of complement opsonization and amplification that we can interfere here, because there we can really specifically block in a time-specific way complement broadly and prevent pretty much all of the effector pathways that complement mediates. So how do we do that? There are different ways we can do it. What we are looking for is this central step of these C3 activation by the convertase. We just heard about from Dimitri, and I showed you a little bit about this something. But the important thing is that C3 has to bind to the convertase for being activated to C3b. And we try to interfere with that. John Lambris in Philadelphia some 20 years ago has discovered an interesting peptide called comstatin, or the family of comstatin, that are cyclic peptides that bind to C3 and prevents it from binding to the convertase, so thereby shutting down pretty much all of the complement activation. And over the past 20 years, and since I also joined the lab, we have been developing that into really an interesting drug that is not only sub-nanomolar binding to the target, but also has high plasma stability and so on. John has founded a company which is developing this latest drug now clinically, but you also have to consider that it has a very narrow species specificity for non-human primates and humans. So that's an interesting compound, but as I just said, this specificity, species specificity restricts us a bit from doing animal models, so we have to do ex vivo human models or going direct into primates. But fortunately, we have a lot of international collaborators that helped us with this tremendous effort, and we were able to test Compstatin in various disease models ranging from local and long-term to short-term, acute. And I cannot cover all of them, of course, but I want to just quickly show an example about biomaterial-related studies. So what we looked at is hemodialysis, for example, and in hemodialysis you have the situation that biomaterial-mediated activation can contribute to inflammatory triggers that these end-stage renal disease patients observe all the time. They have chronic inflammation, and complement activation can contribute to that. So even there were improvement in the filter technologies, we still see that complement can be activated by these even modern filters, even activate immune cells and markers of coagulation and inflammatory markers. So we have been showing that ex vivo. We also wanted to look at that in an animal model. So we had a non-human primate model that was very close to the clinic, where we exposed the monkeys to pediatric filter and equipment, even under hyperinization, a clinical condition, and could show that even if we used a high biocompatible filter, we were inducing complement activation during this 4-hour treatment period. And we looked at different ways to prohibit that. And what we found that a single injection of Compstatin at the beginning of the session was able to completely shut down complement activation during hemodialysis. And shortly afterward, it was cleared again from circulation. We also looked at inflammatory markers and could see that we didn't see a lot of inflammatory markers popping up, but IL-10 as anti-inflammatory cytokine was coming up. So that could be an extra therapeutic effect that complement CB40 can mediate. So we have an interesting model. We still have to see how it translates into the clinical situation, but this is an example of biomaterial-induced complement inhibition. Another one that I'm just rushing through is an implant model with hernia repair meshes, polypropylene meshes that you use to close a hernia in patients. And if you incubate that in plasma, we can again see that Compstatin can shut down the complement activation by this mesh in plasma and also in whole blood. And we can also see that, again, if we expose the mesh to whole blood, that we can also suppress the activation of immune cell by Compstatin, but also by one of these inhibitors that act downstream at the C5a cell activation pathway here. So we transferred that into an in vivo model in mice. We couldn't use Compstatin because of species specificity, but we could look at the C5a receptor antagonist and could again show under therapeutic conditions that we could eliminate this or reduce this activation of immune cells by the biomaterial. So there is 2 examples how we can deal with complement in solution, and we're also looking at different ways of doing that directly on the surface. And we focus on factor H as one of the complement regulators. And what we could show is, even though biomaterials cannot recruit factor H to the surface and shut down complement, we can look for peptides, for molecules that can do that for us. And if we coat the polymers with factor H binding peptides that we discovered in our lab, we can recruit this host regulator to surfaces and inhibit plastic-induced complement activation on these surfaces quite efficiently. This can also be translated to cell models. We just have a PEG anchor here and could show that we can recruit complement to cells and shut complement activation down here too. And think that's a really elegant way of protecting biomaterials in addition to just inhibiting in solution. So what I wanted to show you, the take-home message is that there's different ways of dealing with complement nowadays. In the case of nanomaterials, we even don't have to talk about safety that much because you can really shut down during these first activation peaks complement system for just a few hours, or you can shut it down directly on the surface that is affected. There is fortunately coming more and more complement inhibitors close to the clinic, and what we hope is that we can really start talking about the real effects of complement in those situations and can test it in clinical trials. What it needs to do is to act complementologists, nanoscientists, and clinicians together. I think this meeting is a wonderful platform to bring people together for that. And with this, I thank you very much for your attention, and also all of my collaborators and funding for these studies. Thank you very much.
Thank you, Daniel. Unfortunately, we are very short in time. The next session starts in 15 minutes, so please ask your questions from Daniel in the break. And we come to the last speaker, Professor Barbara